So this audio lecture is going to cover information that's in Chapter 1. Chapter 1 is an overview of the immune system, so it's going to touch upon a lot of different concepts, some of them that we'll revisit during the course of the semester. So it's really just meant to introduce you to some of the terminology we'll be using, and then some of the concepts we'll explore. So if any of this goes over your head, don't worry about it. We have all semester to learn about these concepts and utilize this terminology. So when we talk about immunology, the term immunity comes up. And so what is immunity? Immunity is a state of protection against foreign pathogens or substances. And so this is where, we might say we have immunity to something if we've been infected before, and then we have immune cells that are going to protect us. This concept of immunity goes back over 2,000 years ago. So in recorded history, realizing that people had protection against reinfection was actually documented in 430 BC. So with the term immunity, we have another term, antigen. So we'll talk more about antigens in just a second. So here you can see that we have two different definitions of antigen. So the definition on the top is from our current textbook that we're using for this course, QB Immunology. And so you can see that this one is much briefer than our definition that's from Genway back in 2001. So this bottom definition is actually from the immunology textbook I used as an immunology student in my undergraduate career. And so you can see that this definition's longer, okay? And so if we look at the Genway definition from 2001, we can see that an antigen is any molecule that binds specifically to an antibody, right? And so the term antigen comes from generate antibodies. So antibodies, and then generate. And so the idea here is that for a molecule to be an antigen, we have to be able to develop antibodies. And you'll learn in this course, or maybe you already know, antibodies are generated by B cells. So it's a B cell response. So the reality, though, is that we have T cells, right? So T cells are an important part of immunity, of the immune response. And so our Genway definition kind of doesn't include any of those molecules that a T cell could see. So when we look at our current definition of an antigen, any substance, usually foreign, that binds specifically to an antibody or a T cell receptor. This is a more inclusive definition, so it allows for response by both B cells, being the antibody generation, but also by the T cell, because we have our T cell receptor being present. But again, keeping in mind that the molecule has to be able to bind to one of our adaptive immune cells, and we'll talk more about those immune cells in a little bit. But just keep in mind that an antigen, that term, it's more geared towards our adaptive immune response, okay? I do want to point out that as the definition from our textbook says, usually foreign, we can have substances, molecules that are present that are not foreign, that act as antigens. Okay, so as you're probably aware, we have autoimmune disorders, and this is where our body attacks our own cells, and so in that case, we'd have an autoantigen. So when we look at immune response, they rely upon this recognition of molecules, okay, and this recognition of molecules can occur in two different ways. So we can have germline encoding, and so this is where we're going to have pattern recognition receptors, or PRRs, and so this is where we would have binding to pathogen associated molecular patterns, or microbial associated molecular patterns. So sometimes they're referred to as PAMPs or MAMPs. MAMPs is the more current terminology, PAMs is a little more outdated because they are plant-associated molecular patterns, and so botanists got mad, and so immunologists said, fine, we'll just call them MAMs. So just if you see those two different terms, because this is not a botany class, PAMs are acceptable, but just be aware that a lot of the literature has switched over to calling them MAMs. So again, this idea is that, in our germline, we have patterns that are found in us, and then pathogens, microbes have different patterns that are found, and so we have cells that are able to determine our own cells, self versus non-self. So they're able to read those molecular patterns that are unique to not us, non-self. The other way in which molecules are recognized are through cell receptors. So more specifically, these randomly generated receptors that are found on B cells and T cells. So this is part of this adaptive immune response that we were talking about with antigens. So later on in the semester, we'll talk about how T cells receptors and B cell receptors are generated, but more or less it's random. The genes cluster together, and so a T cell or B cell will randomly pick different sections and then create their receptor. That receptor then, if it binds to an antigen that is non-self, it gets selected for. So again, this bottom portion is actually 4 chapters in our textbook. So just there's more to come on this. So based on whether this is a germline response or whether this is because of receptor that are randomly generated, we can break the immune response into 2 different systems, the innate immunity and adaptive immunity. And so just keep in mind that these, though we'll talk about these as like 2 systems, they're actually interconnected and work together. So to have a good adaptive response, you need a good innate response. Okay. Also keep in mind that, as scientists, we like to name the same thing with lots of different terms. So sometimes you might see innate being called non-specific. And again, this is because this is geared towards those pathogen molecular repeats. So those MAMPs or those PAMs, so it's geared towards that, the recognition of this is not me, so non-self, so must be foreign. With adaptive immunity, this is our T cells and our B cells. This is where we have specific binding to those receptors, and so sometimes it's called specific immunity. So again, just keep that in mind that there's, in this course, we'll talk about innate and adaptive, but if you're looking at a different textbook, like maybe your A&P textbook, or you've taken A&P and you talked about the immune system, innate might have been called non-specific, and adaptive might have been called specific. Okay, so just keep that in mind. So this is a very nice table as you're trying to kind of do an overview of the innate versus the adaptive system. So your innate response is going to be your first response. So the response time is minute to hours after being exposed to something that would lead to an immune response. So if you're exposed to a pathogen, within minutes or hours, your innate response will occur. Where your adaptive is slower, so it's going to take days before it starts to respond. When we look at specificity, innate one is going to be more limited and fixed, and so again, it's just looking at, is this not me? So are there these molecular patterns that are non-human that we should respond to, or not? You have your own barcode, your own ID, and so there's that looking, do these cells have Dr. Schreiner's ID or your ID present? With The adaptive response, when we look at specificity, it's highly diverse, so it's going to actually adapt and improve over the course of time, and we'll look at that when we look at the adaptive response, is how you have a first response versus a secondary response, and with each exposure to antigen, to pathogen, those cells actually are able to get better at having a response. And so when we look at this, the next category here, our next row, response to repeat infection, it's the adaptive response that's changed due to repeated infection. It's more rapid and more effective, where with the innate, it's going to be the same each time. So, if you like to eat contaminated meat, each time you eat that, you're going to have the same response from the innate. Okay, where with the adaptive, this is where you would end up developing a T and B cell response. You'd develop antibodies. Those antibodies would be able to bind and recognize, the E. coli that's in the contaminated meat much quicker and help with that response. Okay, when we look at the major components of innate, we have like our first line of defense, our barriers, our skin, right? So our skin is very much part of our immune response. A lot of the characteristics of the skin, the fact that it has an acidic nature, it has a higher salt concentration. We have defensins that are secreted. We have oil glands that are present. All those different features of the skin actually help to protect us against pathogens entering us. And we also have mucosal layers that are really important, that all act as barriers. And then we have cells, our phagocytes, that are actually going to go in and engulf any kind of foreign material and foreign cells. So phagocytes are immune cells that can undergo the process of phagocytosis. So when we talk about the innate response in our next unit, we'll talk in more detail about the process of phagocytosis, but just know that phagocytes are cells that undergo the process of phagocytosis, so they're part of the innate response. And what these phagocytes do is look for those pattern recognition molecules, so these repeats that indicate that this is not you, again, non-self. Now, when we look at adaptive, the major components would be our T cells and B cells. So these are going to have antigen-specific receptors, so our T cell receptors and our B cell receptors, and then the antibodies that the B cells produce. So first we'll look a little bit more at the innate response. Again, not as in-depth as we will when we're actually, focusing on the chapters that cover innate. So again, for the innate response, it's important to keep in mind that there's the first line of defense. So these barriers are created. So this gives it a fast response, but again, it's non-specific. It's not that, your gut, your innate response from your gut knows the difference between E. coli versus Salmonella. It just knows, it's not supposed to be there, or it's not supposed to be there at those levels. Like, this is not me, and we need to start sounding the alarm and taking care of that, right? So it's going to use germline encoded recognition molecules, again, those MAMPs, PAMPs. And then again, one of the major components when we look at the cellular components of the innate response are those phagocytes, so leukocytes, white blood cells, like your neutrophils, that help to engulf bacteria. Okay, and again, we'll talk more. Chapter 2 talks more about the different cells that are involved in immune responses. Now, again, to compare this with the adaptive immune response, this is where we're looking at responses that are called humoral. So humoral immunity, meaning fluid. And so antibodies are found in the fluid, in your blood, in the serum. And so humoral immunity refers to our B cell response. And then we have cell-mediated response or cell-mediated immunity, and this is our T cells. Okay, so our T cells are going to give us the cell-mediated. So our T cells are going to, go out and fight depending upon what type of T cell they are. So with adaptive immune response, with these lymphocytes, we actually have to select for them, and so it's slower to develop. And so it takes 5 to 6 days in order for B cells and T cells to get selected for, and for their receptor to be generated. So as mentioned, these receptors are going to be randomly generated. So, if we think about the different combinations, because of the genes that are present, we can have millions of different possible receptors. And so that leads to a highly specific response, so that it is directed towards a specific antigen. And so for your adaptive immune response, it could tell the difference between hemorrhagic E. coli versus your run-of-the-mill E. coli that's not going to cause you any issues. So, most of us have E. coli in our gut. It's actually responsible for producing vitamin K for us. So it's certain strains of E. coli are actually very beneficial. And so our adaptive immune response can actually see the difference between them because it's so specific. So the flagella protein is slightly different between the different strains. And again, those antibodies and B cell and T cell receptors are specific enough to see the difference between those flagella proteins, even though, we wouldn't be able to see them unless we actually genotype them. Okay. So again, it's really important to keep in mind that these two branches or systems of immunity work cooperatively together. So, the activation of the innate immune response produces signaling molecules like cytokines, and without those cytokines, you're not going to get the development of your B cells. And so it also helps to direct. So if you need a certain cell type, so maybe you need T helper cells more than cytotoxic T cells. And so these cytokines help to push and drive what type of adaptive immune response you'll end up having. And so it's not an either or. For functional and robust immune response, both have to work. And so those terms, functional and robust, immunologists love those terms. So you'll hear me throughout the semester talk about a functional and robust immune response. And that's, the goal. If you want to have immunity, you need to have a functional and robust immune response. If you want a vaccine to work, you need a functional and robust immune response. And so it's the two, adaptive and innate, working together. So I've already mentioned this idea of self versus non-self, and so the term in immunology that refers to this is tolerance. So tolerance is where immune cells will be selected for if they do not recognize self. And so this ensures that the immune system avoids destroying host tissues. So keep in mind, as I mentioned, T cells and B cells are going to randomly generate their receptors. And so because this is random, it's possible that they could actually generate a receptor that recognizes self. So it would be against self or anti-self. So if this T cell or B cell was allowed to go out in circulation, it would actually start to destroy your tissues. And clearly, as autoimmunity exists, that does happen, but we want to try to prevent that from happening. And so tolerance, mechanisms of tolerances are ways in which we can stop that from happening. So this is where a T cell or B cell that does recognize self isn't allowed to go into secondary lymphatic tissues. So it gets destroyed before that. If it does escape, there's other checkpoints that if a T cell responds too strongly to something, that might indicate it's actually responding to self. So that cell might end up getting destroyed. So we'll talk about the idea of tolerance kind of throughout this semester, but definitely when we're talking about B cell and T cell development. So the immune response is generated against pathogens. generally. And so throughout the course of the semester, we'll talk about pathogens. And so when I use the term pathogens, I could be talking about any an organism that falls into any one of these categories. So it could be a virus, a bacteria, fungi, or a parasite. When we're talking about parasites, we're talking more specifically about eukaryotic parasites, either protozoans, which are single-celled organisms, not, fungi have their own category, but also multicellular organisms like parasitic worms, okay? So viruses and bacteria are parasitic also. Some bacteria is not parasitic. Some bacteria is actually beneficial. And then some fungi are also beneficial, but some can be parasitic depending upon the location they're found in. Okay, so we do want to keep in mind for an immune response against an organism, it can depend upon what structures are found on the pathogen and also its location, both whether it's intracellular or extracellular, or whether we generate an immune response. And so throughout the semester, especially through those journal club papers, we'll look at immune responses to different pathogens. And so there are different mechanisms of response. So how you respond to a virus is different than a bacteria, than a parasitic worm. And that'll become clear when we talk in chapter 2 about the different cells. So we have cells that are good at parasitic worms, so the xenophils. An example of a white blood cell, a leukocyte, that is good at fighting if we have a parasitic worm, where neutrophils are really good at fighting if we have a bacterial infection. Okay, so the immune system's kind of tailored how it responds based off of what type of pathogen or organism is present. So as I mentioned with adaptive immunity, we have both humoral immunity and also cell-mediated immunity. Humoral immunity is where the pathogen is recognized by antibodies. So as I mentioned, antibodies are going to be produced by B cells. And antibodies can actually be transferred from individuals to provide passive immunity. And so you've probably heard this in the news, where if you have recovered from COVID, the American Red Cross and other organizations would like to find out if you have antibodies against COVID, because they're finding that if they do a either blood transfer or antibody transfer, that patients can be helped. So some people have antibodies that are able to aid in recovery for individuals. With cell-mediated immunity, this is where the T cells, the T lymphocytes are involved. And so there are many different subsets of T cells. And so some of these can help to eradicate the pathogens. Other ones can help other cells to induce immunity. So your T helper cells can actually help your B cells, so that they'll produce antibodies. So just like innate and adaptive are interconnected, humoral and cell-mediated immunity is interconnected. So for adaptive immunity, it relies upon surface receptors being the B and T cell receptors. And so these get generated by gene segments that get rearranged. So in the B cell and in this T cell, there are going to be these gene segments, and depending upon what segments are utilized, that would give you your different receptors. Again, we'll talk more about this later on in the semester, so if this doesn't make sense, don't worry, don't worry about it. This is one of these concepts that is covered through multiple chapters in the textbook. With B cells, what they'll do is their receptor can actually become an antibody or an immunoglobulin, and that's specific to the antigen. Okay. So for T cell, the other piece with T cell receptors is that it needs it can recognize its antigen or specific peptides on an MHC molecule. So the MHC molecule would be found on a different cell, and so another immune cell, or even another cell in your body except for red blood cells. And so this would allow the T cell to recognize it. It's kind of like they're talking the same language. A T cell receptor and an MHC can speak to each other. So this figure just shows you an overview of the adaptive response. So here we have this mouse that's being exposed to foreign proteins, infectious agents, and so we can have a humoral response. And if we look on the left panel, we see our B cell with the B cell receptor. Once the B cell is able to recognize the antigen, it'll become a B cell that will secrete antibodies. Those antibodies then can bind the pathogen so that we get elimination of the pathogen, or antigen elimination. So obviously there's a lot of steps along the way. So again, what we'll be talking about later on in the semester. This mouse also will generate cell-mediated response from the T cells. So here on the right panel, we can see our T cell that has our T cell receptor. It would recognize the antigen, and then we could have cytokine secretion. So what we see on our blue T cells, those cytokines then can cause other T cells to become activated, and they can actually go and kill infected cells. So this is where our blue T cells are a little bit different than those red T cells, but they're gonna aid and work together to generate the response, just like the T cells. The blue is going to work and help the B cell end up generating antibodies. So again, these are complex processes. So though we just see an arrow here, keep in mind that there's multiple steps going through this process. And again, why there's 4 chapters just on this process. Okay, so again, if this is one of these concepts that are you're like, I don't understand it, it's okay that you don't understand it yet. Just know that we have humoral response and cell-mediated response. Built into this response, we have a process known as clonal selection. And so clonal selection allows for a number of different individual B and T cells to be developed, but then an individual clone, a single cell, the best cell, actually gets selected for and expanded. Okay, and so once that T cell or B cell interacts with a specific antigen, it becomes selected and activated. And once activated, that's going to cause proliferation. And so there'll be a large number of copies that will have the same T or B cell receptor on it. Okay, so that's what we see in this figure. Again, we have the stem cell. We have our first kind of round of proliferation, which is going to give us lots of different receptors. So each, 1, 2, 3, 4 have all different receptors. Now, some of these receptors might recognize self, so they actually get deleted. So in this figure, 1 and 4 get deleted for whatever reason. So they're not allowed to go into circulation. They were only in the primary lymphoid organ, like the thymus, but because they have a quote-unquote bad receptor that recognizes self, they're not going to be allowed to go in circulation. So for this example, 2 is the good receptor because it recognizes this antigen from this microbe, and so now that causes that T cell or B cell, that lymphocyte, to proliferate. So we expand numbers. From there, we can further kind of customize the response based on the cytokines being produced, so that some of these become memory, some of these become helpers, some of these become cytotoxic. So they can have different functions from there, but they're all going to have the same receptor. So as I mentioned, with adaptive immunity, you get this primary response versus secondary response, and so that each time you're exposed to the antigen, to the pathogen, the response gets quote unquote better. Okay, so this is a nice figure to be able to kind of talk through and understand. And again, if not, for this first unit, definitely by the end of the semester, you should be able to understand what this figure is trying to convey. Okay, so for our primary response, this is when we're initially exposed to antigen, our first exposure. Okay, and so what we're seeing is first in that light blue, we get this innate response. So we're going to get an increase in that immune response, and this can be measured by the number of cells that are present that play a role in innate. And then after that, we're then going to get our adaptive, our T cells and our B cells respond, but this takes time, like we mentioned, like 5 to 6 days, and so we don't see that starting to increase until around that time point, and that's in the darker blue. Okay, you can see towards the end of that primary response, it's more the adaptive response that is going to be present. Okay, and then if we get a secondary exposure, so a repeat exposure to the same antigen, that adaptive response comes quicker. Okay, and this is because we actually have memory. Okay, so for the adaptive, if we have a T cell or a B cell that has a good receptor and it's seen antigen, like T cell 2 there, or lymphocyte 2 there in the last figure, we're going to want to keep some of those, because if we've seen the pathogen, if we've seen this antigen once, there's a good chance we might see it again. And we don't want to have to start from the beginning, right? Because that takes time. We want to keep some of those around. And so that's what memory is. And that's a really key part of the adaptive immunity, is this memory population. Okay. And this happens naturally when you become infected with a pathogen. So can we actually take advantage of this memory population, and can we induce this without actually getting the disease? And the answer is yes, right? So this is the same premise of vaccinations. So vaccinations would be your primary exposure, so that if you're exposed to the pathogen as your secondary exposure, you would have protection against them. Okay, in some vaccines, you need to have multiple doses in order to get enough of an adaptive response and good T cells and good B cells in order to have that protection. But the idea is that you would then have a long-term immunity because of that memory population. And so, again, this is one of those, COVID pieces where you hear lots of information in the news about vaccine development against COVID, because COVID-19 is this novel strain of coronavirus. So yes, we have coronavirus vaccines in like dogs, but those are against different strains, and so we kind of have to look at what's different between these different strains and develop vaccines from them. And so we can see that if we look at different vaccines and look at the number of annual cases before vaccine versus after vaccine, vaccines are able to dramatically reduce the number of cases. Are vaccines 100%? No. So that's also where we need to make sure that we have other treatment mechanisms, such as antivirals and different strategies, such as rotating patients on prone to help with oxygen flow, especially, again, more so when we think about COVID, but even like something like influenza, we have a vaccine for flu, but sometimes the strain doesn't match up to the vaccine that's developed. So all those things need to be taken in consideration when we're looking at vaccines. Later on in the semester, that last unit, we can talk more about vaccines. We can also take a journal club paper and make that about vaccines. So there's actually a really good one about dengue fever that I've utilized before, and it looks at the different different techniques of developing the vaccines. So lots more information about vaccines that we can talk about during the semester. So we want to keep in mind with anything biological, there's the good, the bad, and the ugly. The immune system is no different. So we can have dysfunctions in the immunity, and again, these are topics that we'll talk about a little bit throughout the semester, but more so towards the end of the semester. So when we look at dysfunctions of immunity, we can put them in two different categories. There's an overly active or misdirected immune response. So things like allergies and asthma or autoimmune disorders fall into this overly active. Or we can have immune deficiencies. So this is where there's either a primary genetic loss of immune function. So someone could have a mutation to their genes that causes them to not have a full functional and robust immune response, or it could be secondary. This is where it's acquired immune loss. So for secondary loss of immune function, sometimes these are viral. So most commonly, most people, HIV and AIDS, that virus actually affects your CD4 T cells. And your CD4 T cells are helpers T cells. They help B cells, they help T cells. And so when those cells are killed by the virus, you don't have those present. And so individuals with immunodeficiencies are more likely to have opportunistic infections, such as like thrush, pneumonia, because they don't have that functional, robust immune response. There are other concepts related to immunology that we usually touch upon with the journal clubs or in class activities and such. So transplants, so with transplanted tissues and organs, rejection is an important thing to keep in mind. And so rejection talks about when the immune response is generated against the transplanted tissue. And this is usually because the matching isn't quite isn't as exact as it should be with that barcode, that self versus non-self. So just recently, there was an individual that had a face transplant. I believe it was like 6 years ago. She got the initial face transplant, and then she has started to reject it. So she just got a second face transplant. And this one, the HLAs are more closely matched. They feel that she'll be less likely to reject the second face transplant. Transplant patients are usually put on immunosuppressive drugs. Immunosuppressive drugs quiet the immune response. That rejection can occur. But then there's complications with immunosuppressive drugs because you're more prone to infections because you've impaired the immune response. Cancer is another aspect that we'll talk about. And so the issue with cancer is these are your own cells. So you don't respond. Your immune system's not very good at responding to yourself. So it's one way that cancer is able to kind of circumvent the immune response. So we'll talk about, and there's usually a journal club paper that we do that involves cancer. A kind of new area that's kind of been added into the immune response is the microbiota. So your commensal microbes that live in and on you that don't cause harm, and what is their function as far as, for immunology, the homeostasis of your immune system, but they also have a function in your metabolic homeostasis. And so if there's a flux or an imbalance in your microbiota, how does that then relate to the immune response? And so sometimes there's increased inflammation. And so, again, this is an area of research looking at the role of microbiota in your immune response. And so this is just showing you a figure on that. So in general, I will have a resource slide at the end of each of the lecture slides. So most of your figures are going to be coming from the textbook, but sometimes there's a review article that I will either include in the slides or post onto our D2L page and Canvas page. And so I always highly recommend looking at those review articles because sometimes they might be easier to understand than your textbook, or in some cases, if you don't have the textbook because it hasn't shipped yet, it can be a way to get the information also. The last slide just shows some of the icons that are going to be used so that this becomes a useful reference page. I did post this as a either as an image, like a JPEG, or as a PDF, I can't remember which one, up onto the learning management system. And so it's useful to have because as you're looking at the slides, these are just some of the common icons and some of the common cells. So in chapter 2, we'll talk about these different cells and what are their functions. Are they innate? Are they adaptive? So I also highly recommend starting to look at this and starting to look at the different cell types so that, especially when you're listening to that lecture, you can start to say, okay, well, these ones are the ones that are innate, these ones are the adaptive, this is what their function is, this is what they're doing, this is what they recognize. Because understanding and having that good foundation for the other units throughout the semester is really important. So it's really important that when I say T helper cell, that you're like, okay, I know what that is, I know what it's doing. If I'm talking about a B cell, what's it doing? If I'm talking about a neutrophil, if I'm talking about a basophil, xenophil, the dendritic cell, that you know what all these these cell types are, and you can say, oh, that's an innate cell, that it can undergo phagocytosis. Oh, it likes to undergo phagocytosis with bacteria. So those, having that strong foundation on the different cells is really important. So I would say start early. Okay, so that concludes our chapter 1. Again, it's an overview, so I try to do a little foreshadowing. So later on in the semester, you'll learn about this. We'll cover this in these chapters. So if you feel overwhelmed, don't worry. It's just meant to introduce you to a lot of different concepts so that we can see where we're going to go.